So, do you know where will cortisol go after its release? Obviously, it's going to bind to the target cells. But how is it going to take place? Let's learn about that. Here's the cell cytoplasm, and here's the nucleus that contains DNA. Well, the cortisol diffuses into the target cells and binds to a cytoplasmic glucocorticoid receptors. These receptors are superfamily of steroid, thyroid, and retinoid receptors. The activated receptor glucocorticoid complex enters the nucleus and binds to steroid response element on target DNA molecules. This is going to do two things. This either induces the synthesis of specific mRNA or regresses the genes by inhibiting transcription factor. For example, nuclear factor Kb or NF-kB. For most clinical purposes, synthetic glucocorticoids are used because they have a higher affinity for the receptor, are less rapidly inactivated, and have little or no salt-retaining properties. Well, steroids are example of gene-active hormones. The steroid diffuses into the cell where it binds to cytoplasmic glucocorticoid receptors, as we discussed earlier. But what happens when there is no cortisol? Well, in the absence of cortisol, the receptor is inactivated by a heat shock protein, which is also known as HSP90. Cortisol itself is going to trigger the release of HSP90, and the activated receptor enters the nucleus where it stimulates or inhibits the synthesis of proteins, which then produce the characteristic actions of the hormones. So that's how cortisol is going towards the target cell and perform its function. Let us move ahead and discuss therapeutic uses of steroids, so stick around.